I'm going to take you through a journey of composting, the basics and some systems using what I call a community-based organics collection model. And what I hope to focus on is how important it is to recover food scraps from our waste stream and use them in a positive manner to help improve the health and vitality of our soils. One of the most important things that we use, and it's the only metric that I really believe in to figure out how much food scraps we waste annually. The national standard is about 150 to 200 pounds of food scraps per year per person. And that is a very reliable number. And so that equates roughly to about % of the waste stream is food scraps that could be recovered and otherwise utilized. In the state of Maine, we actually have developed our own food recovery hierarchy. It doesn't differ tremendously from the EPA model, but we specifically want to always reach towards helping those that are food insecure first whenever we could. But even before that, if we can't make waste in the first place, that we'd be much better off. But once that isn't met, we feed hungry people. And then if the food is perishable but still edible, we feed it to animals to improve their health and vitality. And then unlike the EPA model, we believe both composting and digestion are on the same level. They're both equally good choices. The EPA kind of puts digestion above because it produces energy and digestate. But in the state of Maine, having compost to be put into our soil to improve the vitality is equally as important as generating electricity to us. So that's kind of the standard we go to. And then finally, the last option is going to be disposal in almost every case. As far as composting goes, it happens all the time naturally. All we're doing when we compost is we accelerate and optimize the conditions to make a naturally occurring process that might take a year or two happen in 6 to 8 weeks, followed by another 3 months of conditioning to make it higher and better use as we go. As far as benefits go, I think the biggest one is that it improves the health and vitality of our soils. It feeds the soil. It provides functioning food web systems. It also helps to reduce the overall physical compaction or expressive drainage of a soil by improving that organic matter and creating a layer that can help attenuate water, but simultaneously can help treat leachate and other items. So that's really, I think, its greatest value, although during drought, especially in California, you might find that the farmers would tell you that its water holding capacity is the best part because they use less water during the drought periods and still have the same success rate. So it's kind of a really cool thing. But as far as the physical standards, it does take a thick soil and make it less compact and easier for roots to penetrate, so it improves the tilth. It also will work really well for a very drained soil like sand, providing that extra bit of particle matter in there to help slow down the infiltration rate and allow some treatment to occur and attenuation of nutrients in the root system. So it really is an excellent thing. And then one of the things that I really think is really interesting about composting is that it also, when added to the soil, helps to improve the health and vitality of the plants. And it can actually make the plants and crops that you're growing resistant to pathogens and other airborne and waterborne type pathogenic organisms by almost inoculating the plant and giving it an extra bit of vitality and waking up its genetic resistance. And they call that induced resistance, and it's quite an exciting possibility that's happening. And then the thing that I like to tell people as well is it's like having an ATM card. It's not a fertilizer, but there are enough sort of micronutrients and other nutrients that are available over time. So it'll actually help augment your fertilizing process and make it better in the long run. So lots of cool things that it can do. We're going to talk about these things sort of in a basic manner, but just to get you through the understanding. But the 5 characteristics that we look at for composting are carbon to nitrogen ratio, what the oxygen content is, the moisture content, and then the particle size, which determines how all these things interact. So the C-N ratio is nothing more than looking at the % carbon available to the % nitrogen. And the reason why that's important is the carbon is the fuel source that microbes need. The nitrogen, yes.
Mark,
yes.
Sorry, I need to interrupt you Yes, hi, it's Mina. I need to interrupt you. I'm getting a message that your bandwidth, I think you're having some issues with your bandwidth. So can you just turn off your video? And I think
Of
that will
course.
help out. Okay, great. Thank you.
Yeah, let me do that right now. Uh, Actually, how do I do that?
Actually, I can do that for you. No
Would you
problem.
please? Thank you.
Yeah.
All right, great. Thank you. So, as I said, the carbon is the fuel source. The microbes use that for food, and that's always the thing we're looking for, because usually what we need to get rid of is the nitrogen, which provides the building blocks for more microbes to happen. And for that to work appropriately, we need a chemical ratio of somewhere between 20 to 30 parts carbon to 1 part nitrogen. Now, people get freaked out about that, saying, "How am I going to make that happen?" And as a general rule of thumb, we do things volumetrically, and a very safe recipe is, if you're using a bucket loader, 2 to 3 buckets full of carbon to 1 bucket full of nitrogen. So we break it down and make it really simple. But we find that if the seed-N ratio is too high, it will compost, but it'll take a very long time. And usually the pile will be too dry. If the seed-N ratio is too low, it won't compost well because it'll probably be too wet. It'll be very smelly. It'll be very odorous. So that balance is really important to keep things at an optimal ratio. Okay, let's see here. So carbon sources include things like leaves, wood shavings, cardboard. I'm a super huge fan of bedded manures, because bedded manures usually already have an intrinsic biological activity. And when food is added, the microbes actually take the liquid nutrients, and it usually kickstarts them, and they get even more active and more aggressive. I'm not a huge fan of shredded newspaper or any newspaper products, given this current status of PFAS. We don't know what's in what. So we try to go with the more naturally occurring material like sawdust shavings, wood chips, corn stalks. We call it corn stover, straw. I'm not a huge fan of leaves by themselves because they don't have the structure and get wet and compress, and that can be a problem to the compost. So instead, what I prefer to do is to blend them with either some grass or another material, get them cooking, then add the food to a biologically active matrix. So once again, it's looking at that biological activity. In state of Maine, we kind of break our waste down by carbon to nitrogen ratio. As I said, the higher the carbon nitrogen ratio, the biologically active the material is, and the more apt it's going to be to really compost super slow. So type 1A tends to be leaf and yard, wood chips, some vegetative material, and a lot of the dryly bedded manures like horse manure, which has a lot of sawdust shavings in it. Type 1B tends to be food scraps. That's the material that we get from restaurants and cafeterias. We get it from grocery stores. It's kind of the pre and post consumer material. And that usually has a seed-N ratio of greater than 15 to 1, but somewhere around that. And then we've got the real troublemakers, which are the type 1C materials, which have carbon and nitrogen ratios of like 2 to 10. And that would be like your chicken manure or your seafood material, like lobster or shrimp or things like that. So we try to find those materials, and then we develop recipes based on the ingredients and their nutrient qualities to try and get that compatible 20 to 30 to 1. And then we change the weight recipe to a volume-based recipe so we can use it with our tractor. So it's very simple to do. We actually teach it at our main compost school, but we're always willing to help with recipes to anybody that needs it. So as I said, type 1A, you're talking about leaf and yard up top, some sawdust shavings. One of the things I like about shavings is they have good structure, so then we'll talk more about what that means. And then leaf and yard. 1B are the pre-consumer materials, but you can also take post-consumer plate scrapings, things like that. So this is really in the realm of community collection. These guys have storage issues. They need to be incorporated the day they're gotten, or they tend to break down very quickly, really protestable, and become extremely odorous. So at a minimum, pre-amending them with sawdust or another material kind of sucks the moisture out, and the odor is really what's in the moisture. If you get the moisture tied up, you knock down 80 to 90% of the odor potential. So that moisture is key. Oxygen Oxygen wise, we want to have a really good flow of oxygen through the pile so that the core gets bathed in it, because the microbes that work with oxygen are super happy and they don't produce any odors. They just produce water vapor and carbon dioxide as their end products. Those that are struggling tend to produce volatile organic acids as a byproduct, and those are really smelly, and those can be a real problem. However, if you take a really wet pile, mix in some dry material, and reestablish the seed-end ratio and airflow, you often will thrive immediately, because the microbes like the volatile organic acids because they're extremely nutritious. So it's just a matter of getting the right balance back, which can be done fairly easily. One of the things I like to talk about is I am a retired paramedic, but the reason why CPR works for us is when we inhale, we take in 21% oxygen. When we exhale 16%, because our bodies only require 5% to operate. And it's very similar for a compost pile. As we turn that pile, as it sucks in air, 21% comes in, but only 5 remains. The rest is lost through the process due to the driving force of the compost pile. So it's kind of an interesting thing to see. And really, aerobic decomposition is the fastest and least odorous way to compost. One of the things that people talk about all the time is that we got to reach these high temperatures kill pathogens. That is to a certain extent correct. And one of the things you need to remember is the microbes are actually feeding on particles by sort of puking out a enzyme and then slurping in the dissolved material. So their enzymatic activity also helps reduce pathogens and also destroys weed seeds. So it's a combination of both. And all the recording that all the thermometers recording is that there's a lot of biological activity, because it's the metabolism and the heat that gets trapped in the pile. The harder the pile, the happier the microbes are, the more optimal the process is. So we tend to track it that way. This just shows an average compost process. You start out at a very low temperature. It generally jumps up to about 104, 105 within the first 12 hours, and usually within 24 hours, it's up to 120, 130, and we're in that thermophilic phase where we like to be there for about 4 to 5 weeks. And then as the material starts to get used up, it goes back into a declining phase, which we refer to as curing. The longer you cure the material, the more high-end uses it can be used for. Moisture-wise, we like to see around 55% moisture. The way we do that is called the squeeze test. You take a sample of the compost, you give it a good hard squeeze in your hand. If water shoots out your knuckles, it's too wet. If you open your hand and it crumbles out, it's too dry. And if you open your hand and it's a moist ball, it's just right. So we don't advocate buying all these tools. We just use our senses. This one is a really interesting schematic. It's from the old on-farm composting manual. And basically all that is showing is where composting occurs. You see the free airspace, you see the particle, and then the black is the water. Where those three interact is where the microbes are working, because they need all three to optimize the process. Another way to think about it is to take a look at particle size. On the top left, you see really, really fine particles. We call that true, in essence, sawdust. That material absorbs liquid like there's no tomorrow. So it's perfect for sopping up leachate and things like that. However, it will compact and be very wet, and it's hard to get air through it. So too fine, you end up absorbing a lot of liquid, but have poor airflow. On the upper right, you have coarse particles. They're going to have excellent airflow. They're going to give a perfect pile structure. They won't collapse. However, they're not going to absorb anything. And usually with a strong wind, it's going to dry out all the moisture, so the microbes are going to cease activity. So what we want is at the bottom, which is a combination of fine and coarse particles. It's going to be absorbent of liquid. It's going to be spongy enough to hold the pile up, and it's going to provide a nice profile for airflow. So all the things that we hope to have. This is sort of a great photo. This is a photo I took during our poultry study many years ago. In the center of the pile, you notice that it's wet and really smoking. That is where we have 450 chickens, dead ones, the mortalities. On the sides, you see the air flowing in. Piles breathe from the sides and the base on the sides. And you'll notice that there's two areas one on the left and one on the right that are very light. And people think that's ash, but what it is actually is super dry. As the pile really cooked in the middle, it sucked air in from the sides so vigorously that it dried it right out. So this is an example of how air flows through a compost pile and it shoots up through the top. We call that the chimney. And that's what you hope to have in a good active pile. That pile hit about 168 degrees and stayed that way for a couple weeks. So I just want to show you some compost systems before we get into a few other details. This is by far my favorite home compost bin. It's made of lobster trap wire. It's super durable. You take a section that is four feet tall by 12 feet long, you roll it into a cylinder, and that becomes your compost bin. When filled to the top, it holds exactly 1 cubic yard. I did research several years ago at the University of New England, and we found that you needed a minimum of 1 cubic yard in order to stay warm throughout the winter. And we're able to achieve it in these bins using horse manure and food scraps from the school, University of New England. So it worked out super well. I've applied it to a lot of schools in Maine. This is just some kids having a good time building a pile. This is the same idea, but it's 3 bins together. And the concept is you fill the first. When it's full, you flip it to the second and you fill the first again. When the first is full, you flip the second to the third and the first. So you're basically, it's still 1 cubic yard, but you're getting some activity. The aeration is not nearly as good as the lobster trap wire, but the physical turning helps to make up for that problem. So these work really well. The lobster trap wire costs like $80 for the whole setup. These may be $500-$600 to build if you build them correctly. This is not correctly. I'm not a huge fan of pallets because they tend to have nooks and crannies that fill up with organic matter that doesn't get composted. But these guys listen to me, and at a minimum, they put in the hardware cloth to take away those nooks and crannies. But still, it didn't work as good as they hoped it would, so it became an artifact. The Maryland bin system is the idea of that 3-bin system, but built to a larger scale. Each cell is 10 by 10 by 10, and they're very inexpensive, and they work really well, especially for some mortality management. We used them at the University of New England. This is an example of our shed. It had 3 bays that were 10 by 10 by 10. We brought in horse manure that was super active, laid a base of about 18 inches. The floor was concrete. And then we put our seal, whale, and other marine mammal parts in it. And then we covered it with more horse manure. We had plenty of grad students willing to help out, which is awesome. And then a final coating of really hot manure as a biofilter and to help insulate. And we let it sit for 2 weeks. Then at the end of 2 weeks, we flipped it to the middle for another 2 weeks, and then to the third bin for another 2 weeks. And at the end of 6 weeks, the material is ready to go into curing. So this system works really great if you have a steady idea of the waste coming in and it's not all at once. One of these systems could be overwhelmed by too much material. But for an understood system of mortality, it works really good. This is by far what most towns do. In Maine, we have about 80 towns that compost their leaves. And we have a rule that allows you to take up to 5 cubic yards a month without a DEP permit to put into your leaf pile. If you're a farm and you get a compost management plan, you can take up to 60 cubic yards per month, and you're regulated by the Department of Agriculture, not the Department of Environmental Protection. And I actually help write those plans for farm sites to give them a chance to work with the Department of Agriculture as opposed to being regulated by our department. A lot of farmers like that choice. So basically with a turn, one of these static piles, you're expected to only turn it about 4 times a year. Most of the time it's leaves and grass. And it's a really simple system, but if they add food, if they're doing only 5 yards a month, we don't really change the ratio too much. But if they wanted to do more, we would have them start out with the leaf and yard, get a good biological base going, and then add more food as they go and turn more regularly. So it is a good option for communities to look at. This is an example of our town in Yarmouth, Maine. From the far right, you see a fresh pile of leaves, and then we're working towards year number 2. You see the pile is broken down. Year number 3, it's pretty much all finished. And then after, usually about 6 months later, the other pile, the last pile, is used for distribution to the public. So it's a really great, simple system. This is another system that you see. It used to be used a lot for biosolids in Maine, but since that law has passed, we don't really have a lot of biosolids composting anymore. But the idea, it works really well for seafood and other materials, is that you basically build your compost mix over a coarse bed of wood chips, and you usually have a pipe or piping system in there that blows air up through the whole base. And then on the outside of the compost mix, you put a blanket of material to act as an insulator and as an odor scrubber. What people have found is that if you use the right aeration schedule, it optimizes the compost process. But if you provide too much air, it'll actually dry it out and slow it down. So there is an art and a science to using an aerated static pile, but they are very efficient because you can butt piles right up next to each other that you wouldn't be able to do with our next system, which is a turned windrow. But this is an example of an aerated base. You can see that they've got a base of wood chips on the bottom and the fresh compost mix goes on top. And then they would cover that with another layer. And that whole system would be aerated for periods of time per day, usually like 15 minutes on every hour, just enough to keep the process aerobic. In the case of turned windrow, which we see mostly along our farms and most of our bigger facilities, you form this long, narrow, usually no more than 8 feet tall, I know 16 feet wide. And the idea is that the material is put in this long row and it's turned periodically to help continue mixing and optimize the compost. This is usually done by taking a thermometer reading at several places along the windrow each day and measuring the 1 foot temperature and the 3 foot temperature. And whenever the difference between those two, on average across the whole windrow, exceeds 20 degrees Fahrenheit, we would then turn the pile. And so it's a very simple process. It does require routine monitoring of temperature, but it's a very gratifying process for a lot of individuals. This is an example of a farm where they layer their material. They put down their bedded manure, so the heavy stuff goes on bottom and the lighter stuff goes on top. And then they turn it with a windrow turning machine, which is no different than an ice auger that you see people using to go ice fishing, but put it on its side. And as it spins, it throws stuff into the air, and then the shape of the turner be a parabolic shape, which is really pretty cool. These are wicked fun. This in-vessel system is really neat. I hate to say this, but this company has stopped producing the earth tub. This is a very cool system for small resorts. It holds 3 cubic yards of blended material, and basically you turn it with an auger and you move it in and out, and then you also turn the top as you see the gentleman holding the handle. Usually within 21 days, the material comes out and then it goes right into curing. There is an aeration system, and any potential odor goes through a little Schaefer cart full of wood chips as a biofilter. These things were pretty awesome. They when they first came out, they were about $6,500 a unit. Then they had to jump them up due to production issues to almost $11,000 a unit. And then they found the market wasn't really attracted anymore. So they actually stopped building it, which is unfortunate. It's a very great system. They do offer this, however, which is basically about 15 of those in a row. And it's called the Earth Flow System. And it's the same setup. There's an auger that goes through and kind of does the work. And there's a biofilter unit, and it does super well. This thing, though, is also about $125,000 just for the composting unit itself. So as we start thinking about these systems and how to set them up, one of the things is that we need to talk about how to site them. Now, by far, outside composting is the best option if you have a really good site and there's not a lot of receptors or problem areas around you, because it allows you to avoid a cramped space, it allows you better airflow, and it allows you more process control because you're not confined by a building. You want to make sure, though, if you're doing any sort of composting and you're going to build a site, you want it to be close to where you're going to have your feedstocks, which is anything that enters the leaves or food scraps or whatever. You want a footprint large enough to handle everything, including the equipment storage, the amendment storage. Amendment is another word for feedstock, but it's the carbon that comes in. You want to have it at the top of slope so there's no run on, because whenever water runs through a compost pile, it becomes leachate. Whereas if you divert it, it just is storm water. Watch for prevailing wind. They always blow towards sensitive receptors, and if find another location before you get too invested, because they'll complain and you'll get shut down eventually. And definitely not in the floodplain. We've had people try that, and all their work gets washed downriver in the spring. So not a good solution. Sites should also be have ample room for storage of the carbon, as I said, provide minimal interference with other operations, and your process should make sense. Because if your operators have to travel all over the place to get the job done, it's never going to work. So one of the things that I try to do is I try to set up an even process flow, which I'll show you in a second. And then prevailing winds are the way the winds blow almost all the time. And if, as I said, if they blow towards a receptor, you're never going to change that. The only way you can change that is to completely enclose your operation and operate a biofilter that has zero emission problems. And it just becomes a financial conundrum to even try and do it. One of the things I'm a real huge fan of, though, is that whatever site you operate on, whether you put a pad down or not, it should have excellent drainage. Because if water ponds there and gets around the pile is not going to breathe well, and it's just going to kill the activity. So we try to always get that storm water away from our piles and as much as possible moving in a direction that's going to be good for us. If we do have any leachate that's going to come off the pad, then we try to put what's called a level lip spreader or a vegetated buffer to absorb that material before it has a chance to get into groundwater. So, and that's something that each state will help you with, depending on where you are. So how do we get started? What do we do first? Well, I produced a couple of guides that are available on my website, our maine.gov/DEP. Also, I hope that my contact information will be made available after this presentation. And if you want either of these, shoot me an email and I'll be glad to send you a copy. They just help you figure out how to get started and specifically what system you might want to use. And I start with simple systems. This is probably one of our coolest schools in the state of Maine. This is the Massabesic Middle School, and they actually take all their food from the cafeteria. They bring it out to a very small windrow of horse manure, and every couple of days they dig into the pile and they drop in their food, and they keep going along the side of it until they go all the way around the pile. When they've done that, they turn it with this little tractor and they feed it again. This process usually occurs over 3 cycles that lasts about 6 to 7 weeks. And then once the material has had another week or two to sit, it goes right into curing. And the stuff you see on the far right bottom is their final compost. And I've tried to talk them into doing things like making little cubic foot bags of the compost, like a plastic bag or a reusable bag, fill it up with compost, put their sticker on it, and sell it for $5 a bag. There's 27 cubic feet in a yard. And if you sell it for $5, you're getting $135 a cubic yard for that. And that can go into field trips and pay for school expenses. And so I try to get this going so that it also makes parents aware that their kids are making compost and they're using the compost at home. So it's a really positive way to adults with sort of a secondarily to the children. And I like it. This just shows you their final product, and the science teachers on the far left, the groundskeeper in the middle, and then the principal on the far right. And once again, it takes a village to do a great job, and that's their finished compost. It looks really awesome. One of the things that I'm pushing here in the state of Maine is called consolidated collection centers. A lot of communities call them residential drop-offs. Maine, we didn't have a lot of this going on, and so we started this model to give people an idea that they can collect food in one area, and if they generate enough of it, there are people inland that would love to have it if they could get it a lot at one time. So it allows us to stage food for removal to a facility that might want to use it if the community doesn't have the capacity to do it themselves. So we call that consolidated collection, and it makes a food transfer station. It works super well, and we're having a lot of success with it. However, in order to set these things up right, especially if you're working with a business, everybody's got to be on board, because our biggest issue right now is contamination. And so we train staff in the kitchen on what can go in there and what can't go in there. Well, simultaneously on the other end, on the service end, we train people throwing their trash away, what can go in the compost bin and what should go in the trash. The idea is to keep the contamination to zero so that the quality of the product is exceptional. And that's what we're shooting for. The following items are targeted, and basically it's everything that's organic. Coffee grounds is a tough one because there's a lot of folks that say don't put your filters in there, but there are a lot of companies that will give you all the grounds they generate, but they don't want to pull the filters out. And I found that the filters are not as problematic as some of the other packaging and other materials. So I can go either way on that, but simultaneously we try to keep stuff out that's going to be a problem maker, like liquids, milk, things like that. They will, in a big pile, they'll work great. But in a storage container in August, they're going to make a problem odor-wise. So we try to keep those out. Pet waste, I am an advocate of composting your pet waste, but do it away from anything that's going to be used for human consumption, and then spread the finished compost around the edges of your property where people don't play. So you're still going to use the material it was meant to be used, but just not in a food crop system or around where children will be playing. And then, these other things, you'd say to yourself, why would I have to tell someone not to put dirty diapers in? But I will tell you that if you don't put it up, they end up in the container. It blows me away, but that's one of our biggest problems of those soiled diapers. This is an example of sort of doing the pre-consumer material. You can see it's all clean stuff. I will advocate that this go to feed animals when and if I can. But if not, then I'll let it go towards composting. But I always am trying to push things up to a higher and better use. This is the post-consumer, and this is an opportunity to educate for service line folks and say, look, at no paper products. We just don't trust that we don't have any potential contaminants. So just clean food scraps. And if you do that, what comes out is going to be clean. But it's a real labor of education and continued reinforcement of that education. Separation wise, it's not that difficult. You just need to be consistent and reinforce. And the feedback loop has to be positive, even when you don't feel good about it. Because if you're negative to people, they'll stop doing it because they just don't need the hassle. So you've got to find a positive way to tell somebody they didn't do a good job. And it's very trying, but it can happen if you think about it long enough. This is a company in southern Maine called Garbage to Garden, and they do residential and commercial curbside pickup. And they started out this business many years ago in this guy's backyard. He's expanded it to almost 5,000 or 6,000, maybe even a little more subscribers that pay him basically $15 a month. And he takes their scraps and he takes them to be composted or digested. And then whenever they want, he brings them a bag of compost. They can ask for it to put in their garden. And it's a once a week pickup. This is another example of getting a little bit higher up where you can actually collect food scraps from a larger setup. These are 96-gallon containers. You'd see these around apartment buildings, or several families could work together to fill one. But it's another way of collecting a bunch of food to get a whole lot of volume. This is Garbage to Garden's sort of residential pickup where they actually have these at Walmarts and other stores. And once again, you can see the beautiful signage, what goes in, what doesn't. And it just lets people know not to do the wrong thing. And I think these educational kiosks are a great way to go. It keeps reinforcing the right message. People have asked me all the time, well, how do I know how to cite these? What do I do? And we try to find a place that's got a lot of traffic flow, but also there's going to be adequate space. If you don't have space, then it doesn't make sense. So what I put together is sort of an idea of what we need footprint wise, vertical clearance, and dimensions. If we're going to do a single cart, we're going to do maybe a 6-yard bucket, or we're going to use a 20-yard roll off. And you can see that the space requirements is not that massive, even for the largest collection. We're only talking about a 40 to 50 foot by 20 feet. So maybe 1,000 square feet. It's not a massive amount, but what it does is it gives a defined area that you can put signage on. You can put fencing around so that way people know that it's not just a garbage can, it's a commercial food collection area. And then you start educating the public before you allow access. A lot of times we'll start these out by having 10 or 20 citizens bring buckets to fill them. Then once that catches on, we might add another 50 or 60, and slowly we grow this, because if you go slow, you're smooth, and smooth is fast. So the key is just trying to keep those nutrients that you generate within the same zip code and making sure that they end up in our soils. That's kind of my mantra for composting. This is an example of a site that from my university I graduated from back in the 80s. We have a compost operation where the students bring the food from the cafeteria. The town owns the site. They bring us from the racetrack. We build a massive, and the students break in and they dump the food in 3 times a week. Usually it's about 7 to 10 34-gallon toters, so it's quite a bit of food. And then when they've gone around the whole pile, the town front end loader flips it and we start feeding it again. So it's the same as that Massabesic Middle School I showed you, but on a much larger scale. These guys do about 100 to 125 tons a year of food scraps. Very, very good operation. So putting it all together, you need to have an optimal site. You need a site that has deep, well-drained soils, a durable work surface, and really good stormwater control. Key to stormwater is it lands on the ground clean, and if you can keep it clean as it journeys through and by your facility, you're much better off. Because stormwater becomes a diversion issue rather than a treatment issue, you're much, much better off. The next thing that you want to do, going there, you also want to have a good durable work surface, but then you want to have your work surface make sense. So I believe in making operators flow like a clock, either clockwise or counterclockwise. In the scenario below, a vehicle drives in by the curing area, or backs in, so to speak, and the first place you see is the waste bypass. Any junk on their truck that's not compostable goes into that area for being taken to the transfer station or landfill. Then they back into the receiving area and offload the material. Often, if the material is putrescible, they offload it onto a pile of carbon to absorb the liquid to take away the odor, and then they cover it with a pile of carbon. We call that preconditioning. And in that phase, that material can sit for 10 days or more before it's actually incorporated into the amendment, because everything is. Then the amendment storage area is where more amendment is added to get that perfect balanced ratio, the 20 to 30 to 1. And then it goes to the mixing area where it's thoroughly blended. If you're going to do a static aerated static pile, you need to make sure it's homogeneously blended, because you don't turn that during the whole process unless there's a problem with it. In the case of a turn windrow, the mix can be a lot cruder because you're going to be turning that several times during the process. But my experience is that the more homogeneous I can blend it at the beginning, the much better off we're going to be at the end. And then, as you can see, you go to the compost area, and the compost and curing can occur in the same location, but you notice that the windrows are in the same direction as the slope. That means the minimal profile to stormwater if it lands on the pad. The rest is going to flow right by the piles. And then when you're done, you put it into the curing area, which also serves as your stockpile area. And people that bring their truck in after they've dropped off their material, they can rinse it out and take a load of compost back, making a backhaul that cuts the transportation costs, because now they're moving material both directions. So it really becomes a very cool thing. And then finally, you want to make sure that you only process what you can handle. The worst thing you can do is be tip fee driven and get this huge tip fee for whatever you take in and just keep taking it because you're making money. I tell people to take the tip fees and put them in the bank as their rainy day fund, and try to only create the highest quality product they can, and therefore only take enough material to produce that high quality. In the end, they'll be much richer because they'll have less problems and less dealings with their regulatory agency. So I think with that being said, I'm at the end. So I'm ready for questions. And let me see, I'll stop sharing.
Great. Thank you, Mark. That was a lot of information. So I'll definitely make that available to folks, and I will certainly include your email so folks can reach out to you as well. We do have a
Awesome.
question, and it's the same question I have too, actually. So someone asked, can you elaborate on acceptability of bleached and unbleached napkins and restroom paper towels?
Yes. So what we're doing right now is we're trying to get communities interested in composting. And one of the things we're finding is that a lot of the napkins are unreliable as to what the constituents are. But more importantly, we're finding that if we allow too much paper into the stream, it gets contaminated right away. So what we're doing instead is we're pushing only your organic stuff. As I said, if it grows, it goes. And I know paper is once alive, but it's been processed. And so we're trying to keep processed stuff out as much as possible. And so it's more of an educational thing. I think as we go forward and we know more about some of these napkins and what they contain, then we can start saying these are allowed in and out. But we're trying to push the people towards collecting their organics without confusing them with what is contamination, what isn't. We just want straight organics.
Okay, so that's what I was wondering about too, but my question was more specifically, I guess, on like those compostable plates and
Yes,
silverware that
yes,
you can get. So when you mention contaminants, I guess these are certain chemicals that you don't want introduced to the pile?
yes. And without getting into a huge discussion of what compostables contain PFAS versus what don't. It's such a hot issue, especially in our state, because we've passed laws where you can't spread biosolids anymore because of PFAS containing. And so for us, it's a much easier thing to say, let's just keep them out till we know more. And compostable packaging is the same way. It's just a lot of the stuff that they put in there to make it hold the food are these PFAS liners that don't let them soil you while you're eating it, but yet it's still compostable. But what happens to the PFAS? And so it becomes just a huge kind of question mark. So we're just trying to steer our boat away from some of that stuff right now. The only one that I have really tested that I trust is bamboo. I've had a lot of success with composting bamboo stuff. That does break down, but I've done all the plastic ones that they are commercially available at Hannaford's and some other places in Maine. I put them into a very, very, very hot commercial composting system that hit 160 for 6 weeks straight. And cups and forks, they got deformed, but they didn't go away. Plates did, napkins did, but once again, we don't know how much plastic they contain. And if you're following Vermont at all, they're actually dealing with microplastics as their big issue du jour. And we haven't even talked about microplastics in Maine yet. But when you pulverize some of this packaging, the microplastics end up in the compost and people are ingesting that. So
Yeah.
it becomes kind of a big question.
Well, I know Jean is going to address some of these contamination issues next week on
Oh,
Tuesday.
good.
So yes, yeah, she's going to talk briefly, I think, about the PFAS, but as you said, it's an unknown, I guess, and then also other contamination issues. Okay, so let's
Yes.
see. There's another paragraph. There's a question. I'm going to read it, but Mark, you may want to open up the Q&A as well, because there's a little bit I have to read. So this is
Okay.
from John Luthard. It says, good morning. I started a managed on-site windrow composting program at my nursery 4 years ago. I cannot utilize any food or animal material because of the attraction of rats. A neighbor adjacent to my brush for chipping pile outside the deer fence has a small 6x6 open pile in which she dumps all her food waste into that. I guess rats are attracted to that all the time. During
Yes.
a session of chipping this past fall, a large colony of rats scattered back to her pile. However, I have had great success utilizing yard and plant waste only. Do the curing piles need to be turned at any time before being spread in the fields?
Okay, so I'll answer the last one first, and the answer is I like to turn them at least once every couple weeks to keep because all curing does is lets the pile naturally sort of slowly finish itself off. But every time you turn it, you help facilitate airflow, and it just makes it better. So yes, I would advocate at least every couple weeks. As far as the food attracting the rats, the reason why her pile has that problem is because she's not properly amending the material before it goes. And I can honestly tell you, John, that if you pull the moisture out of the equation, the rats will not be attracted because rats don't like horse manure. So when I compost in horse manure, we don't have rats. Because we bury the material, we thoroughly work it in, and all it smells like is fresh horse manure. So really anybody can compost food as long as they do it correctly. And if they do it incorrectly, rats will come. And the sad part about rats and seagulls is once they come, they'll keep trying to come. Even if you get really clever, because they know a food source is in there, they'll just keep trying. So it's just doing it right to begin with.
Oh boy. So you're saying that the right way to do it then is to bury it, similar to what you were describing for those school composting systems. You just kind of go around the pile and you
Yeah,
just
yeah.
bury it. Okay.
It's called pre-amending, preconditioning. The moisture is your enemy. And the biggest place I have complaints is my lobster composting operations. It's because someone will put them in a pile, but they won't properly amend it, and the juice will come out the bottom, and animals are attracted to the stench. And so it's really managing it well to begin with, taking away that leachate, taking away the moisture. You take away the attractiveness to vectors, rats, and things like that.
Okay. All right, we have another question more about curing time. What is the typical duration and what is the overall typical cycle / duration from start to finish?
All right, this is a great question because Gene and I work as subject matter experts for the USDA at some of these poultry situations where there are real huge issues, and so we call the process. If you're going to use the material right away, like from these farms, they can use it right after it's met our 28-day cycle because it's a nutrient-rich source. However, if you take material and you mix it and 28 days later you go to use it and you put it in a flower garden, it's going to kill everything in the garden. So every level of compost has a use somewhere. I can put fresh hot compost on corn ground and it does wonderful. Or I can let it cure for 6 to 8 months and put it in a greenhouse where it does exceptional. So it all depends on what you want for your end use, but curing can be as short as 6 weeks, but I like to see it 3 to 6 months. And I do agree with what Gene said. I mean, we call it cover as well, but in the case of what the students are doing is the pile itself is an active hot pile. When they dig in, they're adding food and they're pulling the cover back down. So it's not being buried, it's just being placed into a hot active pile. And then as we go around it, by the time we get back to where they did that first hole, we turn the whole pile. So it's a simple way to get the community to compost. But yeah, so
Okay.
bury connotates putting it in the ground, but I put it into the pile with cover over it.
Okay, so it's like you're feeding the microbes basically. You're giving them
Exactly.
where they are at the most
And
active.
that's
Okay.
the key to biology. You make the microbes happy and your pile will sing.
Okay, we have another question that came in. If I need to add nitrogen in the form of urea or fertilizer, a nitrogen-based fertilizer, how do I estimate the ratio of C to N?
So what I do with this one is it depends on the size of your pile. I'll go to a lot of communities where they'll have a leaf and yard pile and it's just not working. And I'll bring in chicken manure to help them out. And let's say the pile is 100 yards, we'll bring in 10 to 15 yards of chicken manure. It's like putting sort of, my friend Mark says, putting pepper on your mashed potatoes. You just put enough to make it taste good, not too much that it tastes terrible. So it's the same idea. And a lot of times folks like Gene and myself and my other colleagues on the main compost team and across the country, we know this based on our experience. So rather than try to guesstimate, it's easy to give Gene a call and say, Gene, I've got this pile and I want to add something to it. What do you suggest and how much should I add? That's the best way to go.
Okay, all right. So another one just came in on C to N ratio. So in addition to C to N ratio as a metric, have you incorporated microbial community assessments to further detail what microbial composition is desired and when throughout the process?
We've had students do that as part of some of their research, and it's really, really cool. The problem is that taking the test and getting the results, unless you have a student with a lab right there, it takes time. So a lot of decisions we make about compost piles, especially early on, are snapshot decisions. So there is a test, and I only say this, I don't work for the company, but there's a test called Sovita that's built by Woods End. It was made by Woods End Research Lab here in Vermont, or Mount Vernon, Maine. And specifically what it does is it measures two factors, ammonia, and it measures oxygen, carbon dioxide. And what it does is you can actually tell where your compost pile is in process based upon the feedback of that 4-hour test. And it does equate to microbial activity. It's not 100% of a correlation, but it helps give us something. So I might use one of those if I have any questions, but normally I can tell by my temperature, I can tell by touching it, I can tell by smelling it. And all those are sensors that don't cost money. The thermometer is pretty expensive, but you get one, they last forever.
Some folks did we had some of their compost samples submitted for analyses, and one of them was the Sovita maturity test. And it was really simple to interpret. I liked it quite a bit, but it's just like you said, if there was too much ammonia in the pile, that means it still wasn't mature enough. Ready.
Absolutely.
Yeah. Okay, so another question. Do you mix the final compost material with the soil ratio, or do you apply directly to the garden?
I like what Cooperative Extension says, and I think it's one cubic foot for 1,000 square feet of surface. And what they like to say is that they're a no-till, right? They don't, the tilling is, it's been in and out depending on who you are, but in most cases, they don't want you to till it. They want you to sprinkle it on the ground, and then they want you to rake it in with a small tool, like one of those 3-prong tools. And so the idea is that you're just putting it down enough and working it into the soil so that it feeds the soil instead of overpowering the soil. You can't grow anything in straight compost. It just won't work. So I'd like to see it use no more than 25% of the blend.
Okay. At least I know for garden soils, they say anything over 10% organic matter is excessive. And
right. Well, the compost is 25%, but that equates to usually about 5% organic matter by the time all is said and done. But all the research that's been done, the gentleman that built Sovita is Will Brenton. All the research that he's done has shown that when you add more than 25% compost, A, you're not getting any more of an effect, and B, you're wasting compost, which costs you money. So he sees anywhere from 15 to 25 as being the most benefit, but above 25%, you're just wasting it.
Okay. All right. I think the last question before we move on to our second presentation is sheep or goat manure good to use?
Absolutely. And I will tell you that any dry material like horse manure, goat manure, sheep manure, guinea pig manure, rabbit manure that has a high amount of bedding to the feces and urine is exceptional. Dairy manure on a packed bed where there's a lot of carbon is also very good, but straight dairy manure is wet and dense, and it can be used to make a mix better, but by itself, it's not going to flow oxygen, and it's just going to be really hard to compost. Okay.
All right, Mark, thank you so much for that great presentation and for answering all our questions. If you will be hanging around for Xinwei's presentation, there may be more questions that come up regarding the various systems, but Emily did share your email in chat, so people will be reaching out to you, and I'll also share it in the follow-up email.
All right, that's great. Thank you all very much.
Okay, so our next presentation is about the microbiology of the compost pile, and we have Xinwei, who is local. She is she's a professor within the Department of Civil Engineering at Stony Brook University. Xinwei, thank you so much for being here and discussing this topic with us. You can go ahead and share your screen, and I'll let you know. Yeah, we can definitely see it.
Great. Great. So can everyone hear me okay?
You sound great.
Excellent.
Okay,
so thank you so much for having me for today's workshop and for this presentation. Actually, I did learn a lot from Mark's presentation. Let me just go back for one slide. So I would like to first introduce myself briefly, so you will see why I'm here and what I'm going to cover for my presentation. So I'm going to talk about decomposition science and ecology. So basically, I'll be talking about these microorganisms and the basic information regarding how they would break down the substrate in the compost, and a brief description about microbial ecology. So basically, my training was focused on the environmental microbiology. And actually, so now I'm working in Stony Brook University. I teach courses on environmental biotechnologies. And also my research interest is focusing on the nutrients removal from wastewater, and also the nutrients and resource recovery from biomass from bio-waste. So with this brief introduction, I'd like to first talk about this. Here is the outline of my presentation. So I'm going to, first of all, get a brief background, and then I will be focusing on this compost microbiology with the three emphasis on the mechanisms. So from the energy point of view and biomass production point of view, I'm also going to talk about the key compost microbes involved during the different phases of a composting, and then we'll briefly mention like how environmental factors change would affect this composting process. And then I will be introducing a few methods, how we study the microbial diversity and also the microbial kind of function in the compost. So when the compost is added to the soil, basically we hope this compost can achieve several kind of functions. We hope the compost can increase the soil organic matter, and it will help to increase the moisture holding capacity. And also in terms of the texture and the structure, the compost can help improve. And also in terms of the soil aeration, we hope the compost addition can increase the aeration of the applied soil, and it will support the soil food web, as well as some side benefit include to decrease the potential plant diseases. So however, I think in the many practices, people will find sometimes the compost would do great things for plant production. And so for sometimes it does not do much. And even sometimes it may kill the plant if you applied a not the optimum ratio of the compost to the soil. So people may wonder, did we add too much of the compost, or we add just too little, so there's not much like a like improvement? Or does it really matter about the compost type, or is there potential like a toxicity or other potential contaminants that we introduced in the compost? So actually, all that will actually lead to the fact that we need to learn or understand that really the biology for the soil that we apply the compost to, as well as the compost itself. So I'd like to kind of mention again the definition of the composting. So I highlighted a few key points here. So the composting is an aerobic process. So we will rely on these certain types of microorganisms, the microbial population, which can biodegrade the organic matter and under a controlled conditions. So in my following slides, I will be kind of emphasizing these quite a few important kind of keywords here. So similar to a soil food web. So here I put this very classic diagram showing you the interaction among different types of microorganisms and other organisms. So basically in the plant root zones, so the humic compounds which is the degradation product from the organic matter, it can be different types of like bio waste. So the humic compounds and also these like woody biomass, and they will serve as the food, or they will be consumed by a variety of different microorganisms, including bacteria, archaea, fungi, like a microarthropods, like a protozoa, earthworms, and so on. So it's very complicated interactions among different species. And this is similar to during the composting process, we are also dealing with all these different types of microorganisms that are already present in our substrate of the compost. So as you have seen a similar kind of diagram that describe the overall composting process. So typically we have three different zones from the initial activation phase. And when the composting temperature was kind of below 40 degrees Celsius, and then when the temperature further increased to between 40 to about 70 degrees Celsius, we enter this very intense decomposition phase where we call it the thermophilic phase. And after weeks, several weeks of these intensive decomposition, we'll finally enter the stable and material composting maturation phase, which can take weeks or months to finish. So basically, this composting process is controlled biological decomposition, which organic materials, they are degraded through the activities of a successive groups of microorganisms. So you can see during the first activation phase, you will see a lot of growth of these mesophilic bacteria and fungi. So we're relying on the microorganisms that are already present in our substrate, such as like manure. You would imagine there are like millions or like tens of millions different types of bacteria. And also for a different substrate, you may have a different starting point of those microbial community. And during the thermophilic phase, so the thermophilic bacteria, fungi, and actinomyces, they will dominate and they will grow exponentially to further degrade those organic compounds. And after the temperature reached back to below 40 degrees Celsius, then the growth of these mesophilic bacteria, fungi, and actinomyces will again dominate most of the microbial communities. So with this overview, I'd like to first talk about these the composting kind of the mechanisms. How does the microorganism really break down these organic matters and really harvest the energy to build up the biomass as well as generating heat? So here in this slide, I just want to zoom in this one of the kind of the composting pile. So you just zoom in to look at the microenvironment. No matter what type of substrate we start with, so when you look at this substrate, it can be leaves, grass, like a kitchen waste, or like a papers. So when we consider them as the substrate, typically in a solid form, let me see if I got my laser pointer. So basically, the microorganisms, they will colonize on the surface of this substrate and will form biofilm. So here I just use different colored kind of the symbols to represent different types of microorganisms. So basically what really happens is I took this small green dot as example. So this can be a bacteria or a fungi. So let's say this is a green Bacillus is a typical species of the bacteria involved in the composting process. So it will couple this reduced compound A and oxidized compound B. So here, the compound A is the electron-rich reducing agent. It can be our substrate organic matter, like for example, like wood chips, leaves, food waste, manure, and so on. So those organic matters, they are energy-rich, or we call them electron-rich. So basically, these reduced form of organic compounds, they will donate electrons. And the oxidizing compound B, so in the composting process, we are talking about oxygen, the O2. So the oxygen will accept these electrons donated from organic matter. So via this biological metabolism, so the oxygen will be reduced back to water, and the compound A will be degraded from polymers to simple monomers, or from more complicated organic matters. So we break it down to a more simplified, simple structure organic matters. So during this process, the microorganisms will harvest energy and also will produce energy during this process. So if we look into how much energy can be produced during this composting process, here I want to give you an overview. So this is we call it the electron tower. So you can see this y-axis lists the reduction potential of different types of chemicals or compounds. So we use this tower to to compare the energy production potential when we couple different groups of reducing agents and oxidizing agent. So here you can see at the top of this reduction tower, that means that compound is more likely to donate electrons and to couple with the compound at the bottom, which is more likely to accept the electrons and to generate energy. So here for composting process, you can see the organic matter degradation actually can generate or produce most of the energy. And when we couple it with this oxygen, which is at the bottom, the aeration, aerobic respiration. So when you can couple these two pairs of the reduction oxidation will generate most of the energy. So on contrary, if you look into other processes, for example, when we use hydrogen, so basically hydrogen is also a byproduct generated from organic matter degradation under anaerobic conditions. So if we can couple these hydrogen degradation with like carbon dioxide reduction, so when we combine these two redox pair, that will lead to the anaerobic digestion. So you will see the anaerobic digestion here will generate less energy compared with the composting process. So in the digestion, most of the energy will be stored in the form of methane. So here is the energy production in the composting. So you can find that when we are dealing with organic matters and when we couple with oxygen, so that is the goal for composting. When oxygen is running out, or if we cannot achieve a fully aerated environment, so there will be other types of electron acceptors available in the environment, for example, like nitrate, iron, sulfate. So basically the organic matter degradation will couple with this rest of other electron acceptors. So that will lead to like a hydrogen sulfide production, which is very smelly. So you will see that your compost pile does not have sufficient oxygen. So this is from the energy production point of view to see that this composting process, these microorganisms are using the organic matter and oxygen to produce energy. And for the energy production, so here I have another mechanism slide to show you this biomass production process. So as we just talk about these different types of the energy rich or electron donors, such as uches, grass, manures, and other types of organic substrate. So when these electrons are donated from the substrate, when it couples with oxygen reduction, roughly 50% of the energy will go to these oxygen reduction to generate water and carbon dioxide. And in the meantime, a lot of heat will be produced during this process. So this would lead to the temperature increase in the compost pile. And roughly about 50% of the energy will actually go to the cell synthesis. So basically you will see these bacteria or fungi or other types of microorganisms, their cell density will increase during this composting process. So those energy were used for the fixing the carbon and the nitrogen for cell growth. So if we look into the major microorganisms involved at each of the composting stage, you will see that at the first stage of this initial activation phase, which typically takes about days or weeks. So when the temperature is below 40 degrees Celsius, so first the mesophilic microbes, they will first kick in to degrade the soluble and readily degradable compounds, so such as sugars and like simple structure organic matters. So here I give you some examples of the bacteria and the fungi species that are involved in this process. There are actually a variety of different species involved, including like Pseudomonas, Bacillus. Those are most commonly found bacteria involved in this initial phase, and also some other types of bacteria species are involved. In terms of fungi, so most of the growth of the fungi species are mesophilic. So I will be talking about different types of fungi species growth during this entire stage. But let's move to the next the thermophilic phase. So during the thermophilic phase, so the growth of the thermophilic, or sometimes we call part of them to be thermotolerant microbes, they will break down the proteins, fats, and the more complex carbohydrates, such as cellulose, hemicellulose, and lignans, and so on. And at this stage, because of this high temperature, as well as the competition among different microbial species, we will also be able to achieve the kind of inactivation of the pathogens, and also will be able to kill the weed disease at this thermophilic stage. So the temperature is actually the key here because of the high temperature, but in the meantime, because of the different groups of the microorganism, some of them cannot really withstand this high temperature, and the thermophilic microorganism, they will compete for the food, for the organic matter at this specific stage. So you can see in terms of bacteria, fungi, and we also have these acetomyces, so they become dominant at this thermophilic stage. So you probably would hear people talk about thermophilic and the thermotolerant microorganisms. So I like to spend one slide to talk about the difference or like why people are using these two kind of different names. So a lot of times people use these two different terms like arbitrarily to describe the microorganisms who can live and survive or grow at a high temperatures. So many of these thermophilic and thermotolerant organisms, they belong to archaea, bacteria, and fungi. So in this classification of the microorganism, you can see bacteria and archaea, they actually belong to the prokaryote, and while fungi belongs to eukaryote. So basically the thermophilic and the thermotolerant fungi, so they would grow at like a high temperature in the thermophilic phase of a composting. However, the thermotolerant fungi, they would grow better at the lower temperatures. So basically at high temperatures, so fungi, different types of fungi, they would kind of perform differently. At the mature or curing phase, some of the thermotolerant fungi will grow more compared with the hydrophilic ones, and they will become more dominant at the curing phase. So at the last stage in these stable and the mature composting phase, so the mesophilic microorganism will again become the dominant species. So they will make the remaining organic matter more stable and suitable for plant use. So in this phase, again, these bacteria, fungi, and acetomyces species, they're all present. So as you can see, some of the species, I list them for all three stages. So basically, some of them, they are present, but during the different stages, their kind of activities will be different. So some of the mesophilic microorganism, they will form microcolonies during the thermophilic phase. So they are still there, although they are not really doing much of the activities. So if we look into these different types of the key microorganisms, so we can first look at these bacteria and how they perform during this composting process. So the bacteria population is mainly responsible for the substrate decomposition and heat generation at the initial phase. So their population will first increase exponentially during the initial stage of composting, as they will take advantage of readily available like sugar, starches, these type of organic matters, and heat is produced by these metabolic activities. And if the conditions are suitable, because it's a controlled environment, when choosing the right configuration of this composting pile, the compost temperature will begin to rise. So here I show you the trend of the mesophilic and the thermophilic bacteria growth. At initial stage, you can see the mesophilic bacteria grows much quicker than the thermophilic ones. And also people, when they look into the different kind of types of these bacteria, we use the gram-positive and gram-negative as a method to kind of describe different groups of the bacteria. So basically, both gram-positive and gram-negative bacteria are dominant at the initial stage. So as you can see, basically how we differentiate these two different types of bacteria is based on their cell wall composition. So for gram-negative bacteria, they have a very thin layer of these peptidoglycan. This is like a chemical which will make these cell wall very rigid to the environment. And the gram-positive bacteria has thicker peptidoglycan layer that will make them more resistant to these environmental changes. So based on the staining process, we can stain all the microorganisms, and by looking at their color after the staining process, we use the light microscope to look at these different color, like the gram-positive ones will show this purple color, while the gram-negative cells will show the pink colors. So the gram-positive bacteria, they are more receptive to the antibiotics than the gram-negative bacteria. That is the a general description, like how we would differentiate these different types of bacteria. And during the thermophilic phase, you will see the mesophilic bacteria, they kind of become to a kind of dominant condition that they do not further increase or to increase the microbial population, while the thermophilic bacteria will take the dominant, they will become the dominant species among all the microorganisms present. So during this stage, so those mesophilic bacteria, so what would happen is people thought, so one of the reasons that they will stay there is because they will form a like a microcolonies at this high temperature phase. So during the mesophilic phase, when temperature is low, so you may see these microcolonies of the bacteria colonize on the surface of the substrate. So when the temperature was higher above 40 degrees Celsius, so the thermophilic species, they will take over, and these so basically for these sorry, let me see. So for these mesophilic bacteria, they will form co-aggregates or like form these like a layer system to provide the population from dying or from the heat wave. So at the last stage, during this maturation stage, so these mesophilic bacteria will again kick in, but you will see the overall population will decrease slightly, and also the thermophilic bacteria, the population may be stable or decrease slightly. That is mainly because we may have some predators, such as the protozoa and other types of like a higher level of the microorganism, they will feed on the bacteria. So this is the overview of how these bacteria population would change at different composting phases. So when we look at the fungi, and we can see how the fungi will react or kind of grow at different stages. So fungi are known as the most important primary lignocellulose degraders that involved in the degradation of very complex polymeric substrates. So basically in the first stage, again, so the thermophilic fungi, they will grow kind of slowly, while the mesophilic ones, the population will increase significantly. However, I also want to mention here, whether these mesophilic fungi will grow well, or they will grow faster or slow, really depends on the moisture. So basically high moisture levels in the compost, they will generally favor the bacteria over the fungi. So it is important to keep the moisture in a certain level that you would not have the bacteria to overcompete fungi during this composting. So basically the fungi, the mesophilic ones the population will kind of keep pretty stable, while the thermophilic one or thermotolerant ones, the population will increase significantly during the thermophilic phase. But due to the, based on the literature or previous studies, people found that basically the thermotolerant fungi, so when the temperature is between 40 to 60 degrees Celsius, they will grow much. Well, but when the temperature is exceeds 60 degrees Celsius, so those thermal tolerant fungi, so their growth will kind of enter the stationary phase. They will not further kind of grow at this high temperature. So a lot of these fungi, thermal tolerant fungi, they will also do a good job when the temperature is decreasing to below 60 degrees Celsius. And then when the composting phase enter the maturation, so those high thermal tolerant fungi, they will further kind of increase their populations until we enter this maturation phase, where the predators, they may feed on these bacteria and the fungi. And also in terms of these, like the interactions among the different fungi species. So when we look into the literature, actually, we do not have a very good understanding about how those different species, they will interact at different stages. Just the overview is this overall trend of these thermophilic and thermotolerant fungi at these thermophilic phase and the maturation phase were documented. So another important composting microorganism is the actinomyces. So this type of bacteria is considered as a higher form of bacteria. They are primarily strict aerobes. So they would form a chains of or filaments, and they are commonly found in many environments. So they play a very important role in degrading the complex organic molecules. So you will see in the initial activation phase, their population does not really increase much, but during this activate intensive decomposition phase, those actinomyces, they would grow significantly, and they also play an important role in the maturation phase, because a lot of these type of bacteria, they are thermotolerant, and they will actually slower their development at this nutrient rich environment. So in this thermophilic phase, the fungi and the bacteria, they are still kind of the dominant species. And when these nutrients, because the nutrient is still rich at this condition, so the actinomyces may not become the dominant species. However, in the later maturation phase, so their growth will become more dominant. And basically, this type of bacteria, they will excrete a range of extracellular enzymes that can be used to degrade the insoluble portion or the polymeric carbon sources. So here are a few photos of these actinomyces. So we, in our final compost product, you probably, when we look closely, you may see this kind of like a spider web-like coating of branches or great-wide filaments. So it is not mold in most of the cases. It's actually a colony of the bacteria called these actinomyces. So as I mentioned, so these type of bacteria, they can break down the cellulose and lignin, especially the building blocks of the plant. So they actually play a very critical role during this composting process. So there are also other microorganisms involved during this composting process, but mostly in this final maturation stage. So for example, like the nematodes, basically they are called round worms, and the protozoa. So these protozoa only account for a very small percentage of the overall microbial biomass. And these two types of these microorganisms, they will feed on bacteria and the fungi. So basically, when I look into these nematodes and protozoa, so there's actually another type of composting called the vermicomposting. So basically, this is the decomposition process using various types of species of worms, usually like the red wigglers, white worms, and also other types of earthworms. So this vermicomposting has been used a lot in some developing countries like in India, Bangladesh. So I saw quite a lot of like literatures. And so this is a process that earthworms, they will ingest organic waste, which can include like scrap papers, like a farmland manure, crop residues, and residues of food and leftovers and yard trimmings and so on. So this process is typically considered as a faster process, and the compost product is used as organic fertilizers. Although we emphasize on these worms, the role of those different species of worms, it is still a combination of bacteria, fungi, and worms, these very complicated microbial community to achieve this composting process. I also found some very useful information that on the website of the North Carolina State University. They also did a lot of work on this vermicomposting. So feel free to check these informations on their website. So then with all these the mechanisms and these microbial community change over time, so I like to spend a few slides to talk about environmental factors impact on this composting biology. So I think Mark did an excellent job describing all these carbon to nitrogen ratios and some of those environmental factors. I just want to mention some of these factors, such as the feedstock types. It actually would affect overall the pH of these systems, and also the temperature. Temperature is mainly affected by the configurations, the environmental factors, and also depending on what feedstock we use during this composting process. And here, I will mention briefly about the potential to do a biostimulation or bioaugmentation to improve the overall composting or shorten the composting time. So for the feedstock type here, we have talked about or discussed about the the C/N ratios. So, and also I want to mention the importance of the C/N ratio impact on this microbial growth. So here is a summary table. You can see for different types of organic materials, you may have a different starting C/N ratio. So the more of the carbon available, so basically that means there are more food for the microorganisms to grow and to generate heat. And also for the different initial substrate, so they may have different sizes. We can chop it to a smaller size in order to have more surface area, so the microorganism can form the biofilm or colonize on its surface, and also can increase or actually decrease the porosity depending on their mechanical strength. And here is an example that the fiber, like wood chips, they are more readily to be degraded by the microorganisms compared with these raw wood chips with a bigger size and with less moisture. So the moisture, as I mentioned, this moisture is important to control the fungi and the bacteria ratios in the composting pile. And in the meantime, if we have more moisture, it may also impact these oxygen availability to the microorganisms. So the higher the moisture level, so you probably in the microenvironment, it is likely that we can kind of deplete these oxygen available. So, and also that would affect this redox condition. So basically when oxygen is not available, so the microorganism will prefer to use other types of electron acceptors, such as sulfate or nitrate, and there's a potential to form these odor issues. So with this kind of the overview of these microorganisms, I would like to spend a few slides to talk about the methods that we use to study these microbial ecology or the microorganism in the compost. So in general, there are two main approaches. One is the conventional culture-based method that we could use, like the either use the tube or bioreactors to isolate and grow the microorganisms of interest in the compost. We can separate these like a bacteria or fungi and to look into their physiology and to study like which one is better, can promote a better composting outcome. However, most of the microorganisms, they are actually not culturable. So based on our current knowledge, only a very small portion of bacteria can be cultured in the lab environment. And also, certain types of the microorganisms you can see, such as the bacteria, firmicutes, and other types of bacteria. Certain of them are more easily to be cultivated under the lab conditions, while others are really difficult to separate them or isolate them from. And also the compost microbiology is very complicated. So another approach that people have been using to study these microorganisms is to use these molecular biology tools. So here I list a few different approaches that people have been using in order to understand the what microorganisms are present and like which ones are more important during the composting. So those technologies, including like clone library and the denatured ingredient gel electrophoresis. So these two technologies have been used a lot for the past decades. I would say like in the 2000 or to 2010. So those are still the dominant like approach people have been using. So these technologies are used to kind of to look at the overall kind of abundance and the structure of the microbial communities present in the compost. And later on, people developed this fluorescence in situ hybridization approach. We call it like a fish. So this approach, we can target a certain functional microorganism, for example, like a specific type of bacteria or fungi. So people developed the probes that can target a specific genes in this bacteria or fungi. Then we can use the fluorescence emitted from this hybridization and to see, okay, how many of those microorganisms are present in your compost samples, or whether they are present or not. And later on, people develop this like a microarray approach where we can use a really like a chip format. We can design different gene probes that can target a variety of different microorganisms that are of interest. So we can use get a compost sample and extract the DNA from all those microorganisms, and to use this hybridization approach and using a very high throughput screening process that we can tell whether those microbes are present or not, or whether those functions are present or not. So recently, meta-omics, such as metagenome and metatranscriptomics technologies, as well as a quantitative polymer chain reaction technologies, have been used a lot in the study of the microbial ecology during the composting. So basically, the last three types of technologies are the most commonly used in the research area when we try to understand this microbial ecology. So here I provide you a summary to look at these three different technologies. I will also give you a brief examples like how we use them to really help us to understand this composting process and help us to design a better strategy. So you can see for the microarray, this is a process we can in parallel detecting of these 16S RNA genes. So basically these 16S RNA genes will represent the phylogenetically what microorganisms are present. So it can tell us if we know the most commonly detected bacteria and the fungi in the compost samples, we can design such a primers that we can see, okay, if those important bacteria are present or not. We can also design some of the functional primers that we can tell whether these like ammonium oxidation genes is present, or certain like the disease depression related plant disease depression related microbial species, their functional genes, if they are present. We can also check if some of the pathogens of concerns, if they are present. So these would allow the identification of microbes from different environments and from different composting samples. Another very useful and actually useful tool that we have been using a lot in my research lab is this called quantitative PCR. So this is an approach that we can really tell the quantity or abundance of certain microorganisms of interest. So we can design a gene primers that we can target certain bacteria or fungi. So we can, by doing this quantitative assessment, we can tell you, okay, in your compost samples, your bacteria accounts for like 40% or or these like thermophilic bacteria, which is responsible for your compost, is like a 10% or 5%, we can give you a quantitative assessment. Another useful tool, or actually in order to have overall picture of your microbial ecology or microbial structure, is to use this gene sequencing approach. By doing metagenomes or metatranscriptomics, we can really tell what microbes are really there at different parts of your system or in the same system at different stages. So that will give us an overview picture of the microbial structure. So basically, how does this microbial ecology, or by using those different tools, can really help us during this composting process? So one important application, I think, is to understand the composting process performance, why it is working, why it is not working, and which like microorganisms are taking becoming the dominant phase with a good composting process. So here is an example I want to show you that during a composting from like raw materials, for example, like wood chips or leaves, which is a good blend of substrate, and there are some key composting microbes that are involved in this plant disease suppression. So after we apply the compost to the soil, so they may also help to suppress some of the potential plant diseases. So the microorganisms who are kind of involved in this process, including some of the competitive saprophytes, facultative plant symbionts, and facultative hyperparasites. So for these types of microorganisms, we can use like the molecular biology tools. We can take a sample and do a fast screening to see what the microorganisms are present. We can identify if those species are present in our composting compost product. And then if we want to further look into the certain functions of certain type of disease suppression potential, we can design this microarray approach. So you can see these different color dots, actually those are the different fluorescence signals. So with these like yellow dots that indicate that that target functionals are either present or has been expressed. So those black dots, those indicate those target functions or target microbial species, they are not present in our sample. And based on this information, we can further design a primer to target those microbial species of interest. We can tell you if your compost does have that very useful functional species and what level they are present at different stages. And so this is one example to show you how this microbial ecology could help to understand the composting process performance. And another approach, as I mentioned, this can help us to design a strategy to really improve the composting performance. So there are a few like literatures people reported that since we know in general some of the bacteria, they have been involved in this thermophilic composting process. So people have isolated some of those functional species. They kind of grow them into a higher kind of density, so they can actually inoculate them into the kind of maybe a small pile of the composting. Or if you have a certain compost product performed very well, you can kind of save some of those products and re-inoculate them back to these new fresh compost. So we call this process a bioaugmentation. So basically, after this bioaugmentation, we could change you can see here there are a few small pie chart. So after you inoculate these target functional species, you can change these final product, the final compost, the composition, depending on the final organic matter, like a molecular weight. So basically, you can improve your final compost product composition, depending on your end uses. So with this very brief overview, so basically these are the summary points from this short presentation. So we learned that bacteria, fungi, and actinomyces, they are the key biological species involved during these composting processes. And I emphasize the importance to have the proper feedstock composition and other actually environmental factors. They may greatly impact the compost biology and also the compost product, because we will change the different, the microbial structure involved in these final compost. And also, we will understand the biological interactions at the molecular and ecological levels will help us to better understand the overall composting process and can help us to design a strategy to improve the compost outcome. So with that, that's my presentation, and I'd like to take questions.
Great, thank you, Shinwei. That was a very in-depth look at the microbial population of composting. It certainly is complex.
Yeah.
There are a few questions, and if you could leave your presentation up, because the first question is about, oops.
Oh, sorry.
It's okay. Yeah, if you could just bring your presentation back up, because there was a
Sure.
question about that table that listed the feedstocks and the different chemical qualities of those feedstocks. If you could go back to that slide. And once you get there, I'll read the question. There it is. Yep. Okay, so the question is, can you explain what the numbers mean? So, for urine, it's 0.8 C to N. For sawdust, it's 200 to 500 in the range. Could you just explain this chart a bit more?
Yeah, sure. So, yeah, I think Mark mentioned this in his presentation regarding this C to N ratio. So here is more these based on these mass of the carbon and nitrogen present in each of different raw materials. So for example, like in the sawdust or wood, because it's mainly composed of cellulose, hemicellulose, and lignin, so it's really carbon-rich materials. There's like a less nitrogen in the organic form present in this product. So I think the ideal C to N ratio of a initial starting matrix, I think there's an optimum range about like 20 to 30. So that will make this, we need to consider when we start the good starting mixture, you really want to achieve to that optimum ratio.
So does that mean the lower the number, then it's going to be higher in nitrogen, versus the higher the number, it's just higher in carbon?
I think just for that particular materials, these carbon to nitrogen like mass ratios present.
Okay, all right, so there's another question here. There's a bit going on with this question, so just bear with me. Are you familiar with any current culture-independent applicable efforts to sequence the DNA of mesophilic microbial community members in the maturation phase as a metric for accessibly or affordably developing designer compost to meet certain needs of products for consumers? So that's the first question.
Well, so I have to say, so since this microbial ecology, this area really develops a lot for the past decade, especially with the development of these metagenome approaches, or, actually, when I talk about this microarray qPCR and also the metagenome, actually the metaomics approach is actually really advancing for the past years. So in terms of sequencing approach, really depends on the different substrate that people use. I know in some like research, like papers, articles, people have been sequencing the compost microbial structure at different stages with the same the substrate. There are definitely some effort, but I would say in terms of the well, you mentioned accessibility and affordability. Again, the metaomics technology for now is still kind of costly, I would say, because of the sample size and the replication and so on. So I think for now, people are just trying to understand at different stages what are the major microorganisms that are doing are the most important, that they can kind of be present at all different stages And based on that information, we can design a approach to really isolate them or kind of enrich them for bioaugmentation purpose.
Okay, so it sounds like it's not really available yet, because this kind of second part of this question is, are there efforts to develop inoculants to boost the impact of microbial activity at these different stages in the pile? But it sounds like it's just too expensive to do these tests right now. So
yeah. So
okay.
basically, right. So basically, as I mentioned, there are some like already known like microbial groups like in bacteria and fungi. So people have learned during the previous experience that they have isolated or kind of, I can go back to the slide that in each of the stage, sorry, let me go back here. So you can see like a bacillus, this is like one type of bacteria people have studied a lot. So there are also different strains and species in like bacillus that people found they can really help to improve the composting outcome. So they can actually use some of the very good composting pile and try to isolate those microorganisms and grow them to a certain level and re-inject them or re-inoculate them back to the compost. But in terms of the using the help of these like a metagenome or metatranscriptome, I think there still work to do between this like scientific fundamental research and also the replication. There's still I would say there's still a gap there that need to be filled.
Okay. I had a question just about the different types of composting. So you have the aerobic and then you have the anaerobic. And it seems like if there's, well, correct me if I'm wrong, if there's too much moisture in the pile, then you kind of remain in this anaerobic stage, or I guess you can get to this anaerobic stage of composting. And so does that mean it will just take forever for you to get to the mature compost because you never enter the next phase? Or I guess I don't fully understand the difference between anaerobic and aerobic. Is one more bacteria heavy and the other one kind of has more a balanced microbial population?
Sure. Let me just go to one of my slides that to explain from this like a tower. So I think moisture has, I think it's important in several different parts. One, for the moisture, I think most of the microorganisms, they have to live with like in kind of with the water, right? So all those reactions has to happen in these aqueous phase. So we need to maintain certain moisture levels. And also the moisture level would affect the abundance of bacteria versus fungi. But in another point, when you have like too much moisture and when you zoom into this compost, like a micro environment, when there's more like water, you may have less oxygen available in that small like a micro environment. And here, when the oxygen is kind of completed, there are also other types of like compound present in this compost in the substrate, such as nitrate, sulfate, and so on. So when the sulfate can be a products from like a proteins or amino acids degradation product. So when these microorganisms, a lot of them can live both in aerobic condition and anaerobic condition. We call them facultative microorganisms. So when oxygen is not available, they will switch their metabolism and they will couple this organic matter with the sulfate as their like electron acceptor, and they will also generate energy. They will have this microbial metabolism going on, but they will generate like hydrogen sulfide or even generate like methane in the anaerobic condition. So that is, we will alternate the route of how the organic matter is being degraded. So some of the organic matters will be storage in the form of methane gas. So definitely it will affect or the final product quality.
Okay. All right. It is so complex.
Right.
Yeah, I'm not knowledgeable of this topic, so that's why I have my own questions. Okay, we have another question that came in. As a farmer, sometimes it is recommended not to compost plants with certain fungal problems. I noticed that compost seemed to naturally contain families of bad fungi at certain temperatures. I can't always throw a ton of plant material into the trash. Is there information on how to to compost to control or kill bad or problematic fungi by temperature?
Wow, that is a good question. And actually, I don't know if I'm the best person to answer this. I don't know if Mark, if you're still here, do you
Yeah,
have
feel free,
comment on
Mark or
that?
Jean, if anybody that, is a panelist, and I know you're compost experts in various ways, feel free to join in.
Yeah, from my perspective, fungi are really important, especially early in the process, for breaking down the very high compounds, the high lignin compounds, things like that. Usually, they tend to back off once you get into the thermophilic phase, and then when things cool down again towards the curing phase, you might see a recurrence. But normally, once the major food sources are broken down, they tend to go away anyways. The only time I see them proliferating is if someone's operating a pile poorly. Either it's really, really anaerobic and it's producing lots of odor, and you might see more fungi there. They like the moist environment, but in general, there tends to be species replacement as the process rolls on. That's my general understanding. I'd like to hear what Jean says.
Sure. I agree with what Mark was just saying there. They do change. The beneficial microbes are the ones that go into the soil when we have a finished compost. And Mark was talking a little bit about finished compost and being finished at different points in time. But we see the plant pathogens, and sometimes they'll just take plants with pathogens, put them in a black plastic bag, put them in the sun, and just kill it all. So they're killing by heat. So those plant pathogens are killed by heat in the black plastic bag. We also can do that in the compost process. So I tend to put all of the, if I have a blight or something like that, I will get those into a hot compost. And then that should take care of all of those pathogenic organisms.
So
Yeah,
it seems
agreed.
heat is the better factor than at killing off those pathogens than like competition from the other microbes within the pile or
Yes.
some sort of predation. Okay.
Yeah. And with bacteria and things like that, we can have regrowth. With funguses, they're in there, they will sporulate. And I don't know exactly how, powdery mildew and some of those work, but I believe that we can kill those with the heat.
Yep.
With the heat of
Okay.
the compost. I mean,
Yeah.
that's the idea there.
And coupled with the enzymatic activity too, there is a bit of that occurs as well.
Right.
Okay.
If
Sorry, go
you
ahead,
want,
Jean.
I wouldn't mind commenting on the aerobic, anaerobic.
Sure, go for it.
So digestion is without air and composting is with air. So when we digest, we generally are using those organisms that don't need oxygen, and they produce gas, and we capture, like in a digester, we'll capture that gas so that it's not going off as a greenhouse gas. A lot of it still goes off as a greenhouse gas, just because it comes off of the manure and we can have those gases released. So we're getting a better handle on all of that. In the compost process, those microbes need to have air. So it's important for those to function with air. And like, you will have aerobic and anaerobic processes going on in the same pile. But because you have the carbon biofilter over all of your carbon, is going to be over all of your materials, all of your food waste and all that kind of stuff. So when I said like berry, I just meant we want to make sure nothing's visible. We want to, nothing's recognizable. We want to have carbon over that. And that will help it'll help, and somebody else probably can explain this better, but it'll help scrub that air so that it doesn't go into the air, it goes into the carbon, and the carbon acts as a biofilter.
Okay.